Welcome to AP Biology. This video is on systematics. Systematics is a discipline that focuses on classifying organisms and determining their evolutionary relationships. The goal of systematics is to create a phylogeny, which is the evolutionary history of a species or a group of related species. Through systematics, we are going to create a phylogenetic tree, a diagram that shows evolutionary relationships between different species. In order to create a phylogeny, we look at fossils. Fossils are preserved remnants or impressions left over from past organisms. We also look at homologies between different species. We look at homologous structures. We also look at molecular homologies. We study things like DNA or amino acids shared in common. Here we see some pictures of different fossils. This picture here on the bottom right is one important fossil. This is of an organism called Archaeopteryx. And this is a dinosaur that has both dinosaur features and bird-like features. In the field of systematics, it's important to be able to determine the time period in which a species existed. We have to look at fossils and be able to determine the age of that fossil. There's a couple different ways that we can date fossils. The first one is called relative dating. Relative dating doesn't give us an exact date, but it tells us the order in which different species existed. Relative dating uses index fossils. Index fossils are fossils of organisms that we know when they existed. So if we find other fossils next to these index fossils, we can use the index fossil to give us the general time span in which that organism existed. One important index fossil is called a trilobite. Here's a picture of a trilobite. There are many different species of trilobite that existed, and we know the exact time span by which that organism existed. So if we look at different strata of rock and we see the different trilobites in the different strata, if we find other fossils in that strata, we will be able to determine the relative time span in which that species existed. By looking at the different fossils in the different strata, we could also tell the order in which different species existed. We know that species located in the lower strata are going to be older, and the species located in the higher strata are going to be newer. We can determine the exact age of a fossil through absolute dating. Absolute dating can also be called radiometric dating. In radiometric dating, we use radioactive isotopes to determine the age of a fossil. The most common type of radiometric dating is carbon dating. Carbon dating utilizes an isotope of carbon. Normal carbon is carbon-12. A radioactive isotope of carbon-12 is carbon-14. You can see here that when an organism dies, there is equal amounts of carbon-12 and carbon-14 in that organism. Carbon-12 is not radioactive, it doesn't decay. So you see here that carbon-12 over time stays at a steady concentration. However, over time, the carbon-14 is going to decay. We can look at the ratio of carbon-14 to carbon-12 to give us the date of a fossil. The half-life of carbon-14 is approximately 5,600 years. That means in 5,600 years there will be half the amount of carbon-14 as carbon-12. You see that right here. In another 5,600 years, there's going to be a half of a half, which is going to give us a quarter. In another 5,600 years, there's going to be a half of a quarter, which is an eighth. So to figure out the exact age of that fossil, we look at the ratio of carbon-14 to carbon-12. If there was 1/8 the amount of carbon-14 and carbon-12, we can determine that this fossil is 16,800 years old. And you could see how we figured out that number. It's simply 5,600 plus 5,600 plus 5,600. In order to create a phylogenetic tree, we need to use the field called taxonomy. Taxonomy is the field of science in which we classify organisms. The scientist who came up with our modern classifying system is named Carolus Linnaeus. Carolus Linnaeus created binomial nomenclature. That's a two-word naming system for different species. This two-word naming system is composed of the genus, which is the first word, and the species, which is the second word. So here we see the scientific name of us, of human beings, Homo sapiens. Here we see the scientific name of the wolf, Canis lupus, belongs to the genus Canis and the species Lupus. Taxonomy places organisms into categories. Here we see the taxonomic categories going from most specific to most general or most comprehensive. We begin with species, then genus, then family, then order, then class, then phylum, then kingdom, then domain. The domain would be the largest or most comprehensive category. All organisms belong to one of three domains of life. Either eukarya, we have eubacteria, or archaebacteria. If you look at human beings, we belong to the domain eukarya. We belong to the kingdom Animalia. We belong to the phylum Chordata. We belong to the class Mammalia, so on and so forth. As you go from domain down to species, you get more and more specific. Here we see a picture of what a phylogenetic tree looks like. You can see that as we go from left to right in this picture, we are going through time from older to newer. You can see all the branching points where two new species diverged from a common ancestor. You could also see the approximate time in which that divergence took place. How do we create a phylogenetic tree? We use a form of analysis called cladistic analysis. Cladistic analysis is going to be used to create a cladogram. That cladogram is then going to form the basis of the phylogenetic tree. In order to create a cladogram, we are going to look at structures shared in common between different species. So we're going to be looking at homologous structures. To begin the cladogram, we use what's called an outgroup comparison. An outgroup is a species that existed before any of the members of the in-group. It's used as a starting point to begin our cladogram. To generate a cladogram, we're going to look for synapomorphies and parsimony. Synapomorphies are characters shared by groups, so they're homologies. To generate the cladogram, we're going to use the law of parsimony, which means that the simplest explanation is usually the correct one. This could also be called Occam's razor. To create the cladogram, we're going to be looking at simplest shared characters and the simplest derived characters. Shared characters are characters that exist before that species evolved. So species are going to share this in common because they inherited it from a past ancestor. Derived characters are going to be new traits that evolve that are going to be unique to that group that make it different from the pre-existing group. Here we see a picture of what a cladogram looks like. You can see that the numbers represent the species. The numbers represent the traits or the characters, and the letters on top represent the species. To begin the cladogram, we use our outgroup. You can see that the outgroup is not actually part of the cladogram. It doesn't contain any of the traits, but it's used as a starting point and it's used as a comparison. So 1, 2, 3, 4, and 5 are the traits or the characters. A, B, C, and D are going to be the different species. We look at structures shared in common between these species. The first character to exist is going to be the character that every single species has. That will be character 1. The first species is going to be the species that only has that character. Character 2 is going to be the character that every species has except for species A. Species B is going to be the species that only has characters 1 and 2. And then we continue this all the way up our cladogram. If you look at character 5 here, character 5 is going to be the character that only the last groups are going to contain. Right? If you look at species D is going to have all the characters that evolved before it. So we use these structures shared in common in order to create this cladogram. The cladogram is going to give us the order in which different characters or different traits evolved, and it's going to give us the order in which different species evolved. When we create a phylogenetic tree, there are different types of branches that could be on the tree. These branches are called taxons. There's a major difference between a cladogram and a phylogenetic tree. The cladogram just gives us the order of the traits and the order of the species. But you can see here that the phylogenetic tree shows us where those branching points took place. So it incorporates time into the cladogram. You can see that in these pictures in the top here, we're comparing different branches on the phylogenetic tree, or different taxons. The best taxon that you can achieve is called a monophyletic taxon. In the monophyletic taxon, you have the ancestor and all the species that evolved from it. So it's a complete branch or a complete taxon. Sometimes you don't have that always. Sometimes you have what's called a polyphyletic taxon. You have two species that you know are closely related. You know that they belong on the same branch. However, we don't yet know what their common ancestor is. A good example of a polyphyletic taxon would be the taxon that has both humans and chimps. We know that we're closely related, but we don't know what the common ancestor is yet. That evidence has not been discovered. Finally, we have a paraphyletic taxon. A paraphyletic taxon is an incomplete branch. It has an ancestor and some of the descendants, but not all. Phylogenetic trees are not always perfect. They continue to be built as new information is discovered, as new fossils are discovered. That completes our video.